All right, today we're diving into something huge. It's a brand new study that's basically given us one of the most detailed genetic maps of breast cancer ever. So let's take a look at what they uncovered.
Okay, first things first, you got to appreciate the sheer scale here. We're not talking about a handful of cases. The scientists analyzed the genetic data from, get this, 9,555 different breast cancer samples. I mean, that's big data in a huge way.
And you know, they needed that massive scale because of the core problem they were trying to solve. See, breast cancer isn't just one single bad guy. It's more like a whole crowd of villains, and every single one has its own unique genetic fingerprint. So the real question is, how on earth do you pick out the ringleaders from all that chaos?
So the very first step is just realizing that on a genetic level, we're not dealing with one disease. We're talking about hundreds of different diseases that all just happen to start in the same part of the body. And if you're going to fight something that complex, you absolutely need a detailed map.
And that map idea? That's really the key to understanding this whole thing. Just picture the entire genetic code of a breast cancer cell as this huge, unexplored continent. You wouldn't just charge in, right? No, first you've got to understand the terrain. You've got to map it out.
So the big question is, how did they actually do it? Let's take a look at the playbook these scientific map makers used to chart out the most important genetic hotspots.
And what you're seeing here, this isn't some simplified cartoon for a presentation. This is the real deal. It's the actual honest-to-goodness workflow from the study itself. This is the blueprint they used to find their way through that just massive ocean of genetic information.
Okay, so that complex-looking blueprint, it really boils down to three main steps. Step 1, they gathered all that data we talked about. Step 2, they sifted through all of it to find the genes that were mutated way more often than you'd expect by chance. They ended up with over 1,000 of those. And finally, step 3, they started to figure out how all these problem genes are connected and how they work together.
So after all of that digging, all of that analysis, they found them, the major landmarks on our genetic map. So let's get to the big reveal, the 5 most mutated genes, the absolute most wanted list of culprits in breast cancer.
And there they are. I mean, look at this. The top 2, PI3CA and TP53, they're in a completely different league. Each one is mutated in more than 1/3 of all cases. That's huge. And right behind them, you've got GATA3, CDH1, and MUC16, each popping up in more than 1 out of every 10 patients. These are, without a doubt, the 5 biggest players on the field.
Okay, this image, wow. This just perfectly captures that complexity we were talking about earlier. Now, don't try to read every little thing. The key here is to just see that every single one of those thin vertical lines is a patient. And every little colored mark? That's a specific genetic error. It's just a sea of mutations, a truly chaotic landscape.
But, and this is so important, buried inside all of that visual noise, there's a clear signal. Out of all those thousands of possible variations, it comes down to just 5 genes being consistently broken in over 10% of all cases. That makes them absolutely critical targets for researchers and doctors.
So, okay, it's great to know the names of the individual problem genes, but the real game changer, that comes when you start to understand how they work together. They do this by controlling the cell's internal communication networks. You can think of them like cellular highways.
So what exactly is one of these signaling pathways? Well, the easiest way to think about it is like an instruction manual for the cell. It's a series of commands that tell a cell what to do: grow, stay alive, or even it's time to die. And a single mutation in one of these pathways, it's like a critical typo in that manual that makes the whole system go completely haywire.
Let's take a look at the PI3K AKT pathway, for example. This is the one that's messed up by that #1 mutated gene, PIK3CA. Just think of this pathway as the cell's gas pedal. A mutation basically jams that pedal to the floor, and the result is what you'd expect: constant, uncontrolled growth.
And then on the flip side, you've got the TP53 pathway. This one's controlled by our #2 most mutated gene. If the other pathway was the accelerator, this one is the brakes. And a mutation here, it's literally like someone cut the brake lines. It disables a super important safety check that's supposed to prevent damaged cells from multiplying.
Okay, so we've got this amazing, super detailed map. What does that actually mean for people in the real world? Well, it means we can now write much, much better guidebooks. And by guidebooks, I mean more accurate, more comprehensive genetic tests for doctors and their patients.
And this right here, this is where the rubber really meets the road. Take a look at those genes in bold. PIK3CA, the single most common mutation found in this gigantic study, isn't even on most standard testing panels. Same goes for GATA3 and CDH1, both found in over 11% of patients. I mean, this study shines a huge spotlight on a major gap between what we now know is common and what we're actually testing for.
And the researchers themselves couldn't be clearer about this. They basically say, and I'm quoting here, given the prevalence of these mutations, it is crucial to include them in BC testing panels. You can't get much more direct than that. It's a straight-up call to action, and it's backed by a mountain of evidence.
So, when you boil it all down, the takeaway is actually pretty simple. This research has given us a map of breast cancer's genetics that's just way more detailed and accurate than what we had before. And that map is our guide, showing us exactly what we need to do to build better, smarter, and more complete genetic tests. Which really leaves us with this final, and I think very hopeful question. With this kind of map, with this level of detail, we're not just fighting a generic disease called breast cancer anymore. We're starting to get to a point where we can fight each person's specific cancer. So you have to wonder, how much closer does this bring us to a future of truly personalized medicine?